we should have a makeup class. You know, we should have makeup to we should have makeup class. Okay. Are you free in Fridays? Are you free in Fridays? Good morning. Because the only time I'm free is in Fridays. Now I have all of their lectures turned into my presence. Yes, yes. They all the students should go to the university, and we have the class in the university. So we don't have any online classes for the Iranian students. Do you want to come here? Do you want to come here and have class in the university? Yes. Okay. No, as I know, students, and for the international students, the dawn is okay, and they fix the problem for the international students. But if you want to come to Iran and have the class in a university, I think it's so better, because you can understand the lecture better, you know. So difference between online classes and the classes which are present in the university. Who wants to come Iran and have the class in the university? Who wants to come here? No? No one? One. Okay, but I will be so happy to see you. I will be so happy to see you and have the class in the university. So, okay, no problem. As I know, the dawn problem is fixed and you can have your online classes. But if you can come, I would be so happy. And what will you do for your your thesis? What will you do for your thesis? You want to come to Iran or do it in the Iraq? Okay, Fatima. I asked you which lesson, which lecture? روش تحقیق. In that time, I have a molecular biology lesson. I told you, as I know, I told you the lecture of Ravish. It's not for me. I had molecular immunology in that time. Okay. I will talk to Dr. Amini, and I will tell you about this class. I will talk to them and I will tell you, okay? Don't be worried. Okay, let me upload the file, please. Please wait a second and let me upload the file. About your روش تحقیق class. Okay, she will answer. When did we finish in the previous session? Do you remember? Where was we? Okay, no problem. Where did we finish in the previous session? Do you remember? Type. Fatima, do you have the book? Okay. The type of the plasmids are in the book page 16. Please open your book to page 16. All of you, please open your book. The example was called Yes. Okay, I'll continue. I told you about the lytic and lysogenic cycles. As I told you, for example, we have for example, like this. Here, lambda, this is lambda, and this is M13 phage. Okay, we have two different. The lambda has head and tail structure, and the M13 are filamentous. Look, it's filamentous. In the outside, there is proteins called capsids, and the inner side is the DNA molecules. So, and where is the DNA for the lambda? The DNA for the lambda is here. DNA for the. I explained that. Please ask the. Explain that. This is the phage lambda. Okay, and where is the DNA in the M13? The M13 DNA is inside. Okay, here it is. This is the phage lambda. The structure of attachment is an infection is like this picture. Look, in the first stage, the phage attached to the bacteria. Look at the first stage. Look here. In the first stage, the phage attached to the bacteria and injected DNA into the bacteria. Okay, so in the first stage, the phage attached to the bacteria, and second, the phage replicated. The phage DNA is replicated. Look at here. So, after the insertion, the DNA of the phage is replicated. What will happen next? In the next, the capsid molecules, look at here, the capsid molecules synthesize. Look here, the capsid molecules synthesize. The DNA and the phage capsids assemble together. So they assemble together. Bacteria, open a bacteria, analyze the bacteria, and come out. Look at here, come out of the bacteria. So this is the stage of infection of phage lambda. We call it lytic. Phase. After that, we can have lysogenic. Here is lysogenic. Look at here. Lysogenic. Here is lysogenic, and here is lytic phase. Look. First, the phage attached to bacteria, and then insert the genome to the bacteria. Look at here. Insert the genome to the bacteria. Look here. This is the lambda DNA. The lambda DNA can go inside the bacteria genome Look at here. It can go to the, or it can insert to the molecular of the bacteria genome, so it wouldn't kill the bacteria. It wouldn't kill the bacteria. We call it lysogenic phase. We call it lysogenic phase. And we call the phage prophage. Prophage. Okay? So what is the prophage? The prophage is the phage which inserts into the genome of a bacteria. Okay? So in the lysogenic phase, there is no killing of a bacteria, and bacteria can stay alive. And in the lytic phase, look at here. Look at here. Can come out and multiply and synthesize the capsids, assemble and lie. It means this is the lytic phase. So the lysogenic phase can go to a lytic phase. I told you prophage is the phage which inserts in the genome of a bacteria. Genome of a bacteria. Do you understand this? Do you understand this? Yes? Thank you, Mr. Haydar. Thank you, Bushra. Thank you, Roya. Fatima. Okay, so this is lytic and lysogenic. If you don't have any questions, we can go and study about phage M13. M13 phage. Look at this. This is M13 phage. Look. Okay. As I told you, in the phage lambda, it would attach to a bacteria and insert the genome into the bacteria. Okay, now here M13 can attach only to a pilus. So if the bacteria pilus, if the bacteria has pili, M13 can attach to a pili So, what is the difference between M13 and lambda? M13 can only attach to pilus, but lambda can attach to the surface of a bacteria. So, the genome inserts in the bacteria and it replicated. Look at here. So many genome is here. And after that, the capsid molecules synthesize, and assembles out of a bacteria, but they don't kill the bacteria. So what is the difference? Lambda kills the bacteria in a lytic phase, but M13 never kill the bacteria, just slow down the bacteria. So M13 never kills the bacteria. They can go through the bacteria, extrude from infected cell. Look at here, new M13 lyses the bacteria. Okay, so they won't kill the bacteria. In the late stage, where did the bacterial chromosome go? They won't go anywhere. Here it is. Here it is. This is the genome of a bacteria. This is a genome of a bacteria. In this picture, they didn't draw a genome of a bacteria. This is the genome of a bacteria. The genome of a bacteria is here. Okay, Iman, understand? The genome of a bacteria or chromosome of the bacteria is here. You're talking about the genome of fish. Okay? Okay, Iman, understand? A rolling circle. The process is called rolling circle. Okay. Look here, everybody listen. Look here. I just want to draw it. We have, look at here. We have circular double-strand DNA. Okay, is it clear up to now? Endonuclease and endonuclease cut one of the strands. So, endonuclease A, one of the strands, okay, and the inner strands plays as a template and circulate. This is the inner one. The inner one circulate and look at here. The outer one continuously grow. Look. At first, the outer strand breaks with the endonuclease, then nucleotides attach here. The inner strand play a role as a template, circulates, and the nucleotides attach here. Look at here, like this. The inner strand play a role as a template, circulate, and the nucleotide attach here, and we have a very large molecules called catenon. So we have a large molecule called catenon. After that, the second strand of the DNA, synthesize. So up to now, the outer strand continuously grow, and the second strand synthesize. After that, we have some places called cause sites. Cause site. What is the cause sites? Cause sites are the sites which can recognize by endonuclease A, by endonuclease A, endonuclease A, okay, and can cut these sites, and after these sites cuts, they can become circular, like this, and after that, the protein of capsids synthesize and assemble, and we have so many phage particles. Understand? Do you want me to repeat? Or no, you understand? Okay, Fatima, look at here. Did you learn about rolling circle? Did you learn about it before? Rolling circle, do you know this process? If you don't know, oh, okay, thank you. Please search the videos of rolling circle in the internet, okay? Search the rolling circle videos. For example, you access to YouTube. All of you can access to YouTube. Yes. Okay. All of you, please search rolling circle. I write it here. Rolling circle replication in YouTube. Okay. And if you see the animation, or film of a rolling circle replication, you can understand it better. Okay, so all of you, please search it in the YouTube and see the film. Please listen to me. I told you we have two strands, circular DNA. Okay. The outer strand breaks with endonuclease. Okay. It doesn't have definition. It's just the name, Iman. It's just the name. Okay. In outer strand, DNA breaks. We have 3'. OH free we have 3' OH3 and nucleotides attach the 3' and the inner strand. Look at here, inner strands play a role as a template, and circulate, okay, like this, and the nucleotides come here and it would grow like this, and it would extend like this, and after that, the second strand will synthesize like this. I understood, but right head back with you, so I need to do until I could write. Okay, right, no problem. I will be so happy to read your feedback. And I told you, students, anytime you want me to repeat, I would be very glad to explain more. No problem. Okay. After that, so is it clear up to now? So we have double-strand DNA, which call catenon. Okay? Which call cause sites. Cause sites are the places which endonuclease A can cut them. Okay? So when the endonuclease A cut the cause sites, it can prepare a cohesive end. It's about 12 nucleotides. Okay? It's about 12 nucleotides. We have cohesive ends. So if we have cohesive ends, they can attach to each other and become circular. Understand? So we have so many circular DNA molecules, and after that, synthesis of capsids and assembles, and we have so many phage particles. Okay? Understand? Thank you so much. This is rolling circle. I recommend please watch the animation in the YouTube. Okay? It would help you to understand better. Let's go to M13 infection cycle. Let's go and talk about M13 infection cycle. As I told you, M13 can only attach to pilus. Look in the previous picture. Lambda can attach to anywhere. Look at here. Lambda can attach to a surface of a battery, but look, M13 only can attach to a pilus. So after the M13 attach to a pilus, they insert their DNA into a bacteria. At first, we have single-strand DNA. Then, after that, it becomes to double strands. So, first, is it single-strand DNA? then it will change to double-strand DNA. We call replicative form. We call replicative form or RF. So, all of you, listen to me. M13 attached to what? M13 attached to? Please tell me. M13 attached to? Pills, thank you, Fatima. Then insert the DNA. Then insert the DNA. The DNA is first single strand. Okay? The DNA is first single strand and then become double strand. We call the double strand replicative form or RF. Then RF, look at here. RF can replicate it and create other double-strand DNA molecules. How? Like that one by rolling circle process. Okay? So, in M13, we have single strand and also double strand circular DNA. What is the double strand DNA? Double strand is the form which is replicative form. If the DNA wants to be replicated, it should change to double strand DNA. Okay? Look at here. And after that, like this, like the process I told you, outer strand breaks. We have a nick in outer strand by endonuclease, and inner strand play a role as a template, circulates, and the nucleotides attached to an outer strand. Look at here, outer strands. And we have circulate DNA and phage capsid particle synthesize. And we have mature phage particle. I have a question. Please answer me. Does M13 kill the bacteria and go out? Does M13 kill the bacteria and go out? No, Fatima, thank you. They extrude out of the bacteria. But they never kill the bacteria, I told you. So we have RF form, which is double strand, which by the rolling circle can replicate it. We have single strand and capsid particles synthesize and extrude the bacteria out of the bacteria and never kill the bacteria. Okay. Do you have any questions? Do you have any questions? No, okay. Thank you so much. You are very good students. Let's go to chapter 3. Let's go to chapter 3. We want to talk about purification of DNA from living cells. We want to talk about purification of DNA. from living cells. I told you the questions was here. I told you if the M13 kill the bacteria. All students said no. Okay, M13 does Lambda kills the bacteria, lyses the bacteria, but M13 doesn't kill the bacteria. Okay. If you don't have any questions, we are going through purification of DNA. Who did the purification of DNA in the lab, in the laboratory? Who did this process of purification of DNA? Anybody works in the laboratory? Anybody works in the laboratory, in the lab, and purify the DNA? No? None of you works in the lab? No, it's not a tough process, Fatima. So easy and routine process. If you works in a laboratory, the first stage of every molecular process is purifying a DNA or RNA. Mustafa, you work? What do you do in the laboratory? What do you do, Mustafa, in the laboratory? So, now we want to talk about purification of DNA. As all of you know, this is not Fatima tough process. This is very, very routine process, and all of you should understand. Explain in this book was complex. No, not complex, Fatima. I explained to you, and you understand. This is very, very easy. Okay, so let's go and explain. Listen to me. The first stage is we should look at here. We have some bacteria, and we should culture the bacteria. We should culture the bacteria here. The first, I tell you all process in one picture, and I explain more in the future. Okay. So, what is the first step? Look at here. The first step is culture the bacteria into a medium. You all did this in your bachelor degree, yes? You all cultured bacteria, yes? What did you do in university in bachelor degree? In lab. Yes? You all culture the bacteria. In which medium? Bros agar. Do you know about bros agar? What is bros medium? It should be liquid. Yeah, it should be liquid. Bros agar is liquid. Yes, Fatima, thank you. So, the first one, everybody listen to me. The first one, we should culture the bacteria in bros agar. then centrifuge it. If we centrifuge the culture of a bacteria, what will happen? What will happen if we centrifuge the culture? The bacteria are heavy, yes? The bacteria are heavy, and if we centrifuge the culture, the bacteria will settle down, come down. Yes? Is it clear? Is it clear? So, at first, we culture the bacteria, and then we centrifuge, and if we centrifuge, the bacteria are heavy and they come settle down, deposit at the bottom. Thank you, Fatima. Yes, very good. And we call it plate. Okay, we call it plate, and we dispose the outer phase. We dispose the outer phase. We call the outer phase supernatant, and call here plate. No, Bushra, this is the whole bacteria. This is not the cell extract here. After that, we call it cell extract. Okay. So, in the second stage, we centrifuge. In supernatant, we have what? We have medium. Do we have medium? Do we want medium? No. We dispose it. What do we want? We want the bacteria. Where is the bacteria? They settle down. Okay? We call it plate. Plate of a bacteria. Okay? So, after that, we want to break the bacteria. If, for example, this is the bacteria, and if the genome is in the bacteria, what should we do? We should break the cell membrane. We should break the cell wall of a bacteria. So after that, we break cell membrane, we break cell wall of bacteria. Okay, the cells are broken. So what do we have? Cell extract. The inner of a bacteria are cell extract. Is it clear up to now? Okay, after that, what do we want? We want to purify the DNA. After that, we want to purify the DNA. What does it mean? For example, if we break the cells, what do we have in the cell extract? Please tell me, what do we have in the cell extract? What do we have in cell extract? For example, we have lipids. Yes? We have lipids. We have proteins. What else? We have, for example, RNAs. Okay, so in the cell extract, we have DNA, proteins, lipids, or polysaccharides. Yes? Do we want polysaccharides or proteins? Enzymes or proteins, Marve, yes? Do we want them? For example, polysaccharides, lipids, proteins. No, we don't want. Do we want RNAs? No. what should we do? We should destroy all these components and only have DNA. Okay, so in this stage we purify DNA and destroy all proteins, lipids, polysaccharides, and all of them. And after that, we concentrated the DNA. We don't want so many water here, so we should concentrate the DNA. This is the a very brief stage of a DNA purification. Understand up to now? Okay, now we want to explain more. Every stage of the every stage of this process. The first stage, I told you, what was the first stage? Look at here. The first stage was culture the bacteria. Yes? Okay. And now, listen to me. If we want to culture the bacteria, we want medium. We have two kinds of medium. At first, you should know, I told you, we want liquid medium. We call it brass agar or bus culture. Okay? We can have defined medium. We can have defined medium. Look at here, a defined medium. What does defined medium means? The defined medium means all the particles is clear. Okay? So, we can have defined medium or we can have undefined medium. What does undefined medium means? The undefined medium means we don't know what is in the medium. So it has so many particles, it has so many components, but we don't exactly know what is in it. Okay? The first, I am talking about defined medium. M9 medium, look at here. M9 medium is one example of defined medium. Look, in M9 medium, this is all particles, this is all components. This is all components of M9 medium with the exact dose. So, defined medium, we know all components with the exact dose. But LB or Loria-Bertani medium, LB medium. In LB medium, this is undefined medium. We don't know the exact component and we don't know the exact dose of any of them. So, look at this. In the Loria-Bertani, it means the ingredients are doesn't know exactly. For example, this is yeast extract. So if we look at here, if we have yeast extract, we break yeast, we don't know what is in it, but it has so many nutrition it has so many nutritions in it that bacteria can use it, but we don't know the exact components. So, for example, we have yeast, tryptophan. Look at that. We have tryptophan. Tryptophan, a lot of amino acids and small peptides. If you want to use amino acids and small peptides, you can use tryptophan. And what does extract provide for the bacteria? This provide the nitrogen requires and sugar, organic nutrients, okay, and organic nutrients for the bacteria. So this is two types of bacteria we use for two type of medium we use for growing the bacteria or culture the bacteria. Which you used in your bachelor degree? Did you use LB medium in your bachelor degree for culture bacteria? Did you use this? Many students in the bachelor use LB because it's so easy. No, you didn't work with them? You didn't work with them? Yes, you did. Okay. So this is two types. What is the difference? When it's better to use in lab, yes, in lab. Please tell me, what is the benefits of each of them when we use defined medium and when we use undefined medium? When we use defined, for example, listen to me, if we want to have a very certain experiment, and for example, we want to add something specifically, and we want to know about what it is in medium, we should use defined medium. Why? Because we want to know what is in the medium, and we want to understand what does our, for example, material do. But if we just want to culture the bacteria in order to grow so many bacteria, and we have so many bacteria, and it doesn't make any difference what is in the medium, it's easier to use LB medium. It's easier to use LB medium. So if you want just to multiply the bacteria and we have want to have so many bacteria, LB is the better options. But if you, for example, I told you, if you want to have very controlled conditions, very precise control condition, you should use defined medium. Okay. And when we stop, when the bacteria grow and we have so many bacteria in our medium, we stop the culturing the bacteria. When? How do we understand? Did you remember in the lab of bacteria, you see the medium. You see the medium. How do you understand you have so many bacteria? You can use a spectrophotometer instruments. You can use a spectrophotometer instrument. As you know, you can use a cuvette and you can add, for example, some amounts of medium, and in the spectrophotometer device, the light go through the medium and transmitted light detect from a detector, and we have OD. We have OD of a bacteria in the 600 nanometer wavelength we use, and if the OD is, for example, 1 unit, we have this amount of cells per milliliter. So each unit presents this amount of cells per milliliter. So we can stop the culturing the bacteria if we have a specific amount of a bacteria. Do you understand? Did you use the spectrophotometer instrument? Okay, please tell me, if the OD is 2, how many cells do we have? If the OD is 2, how many cells do we have? Yes. We have 1.6 plus 10, 9 cells. Okay? Duplicate. Thank you. So, this second stage, you don't have any questions? You don't have any questions? Okay. Thank you. So, you have many questions. Okay, ask me, no problem, Fatima. I repeat very briefly, as I told you, if we want to extract the DNA, if we want to purify the DNA, at first we should culture the DNA. We should culture the DNA. Yes. At first I told you we should culture the DNA. Which medium? Brass agar. Which medium? We can have 2 defined or undefined medium. If we want to have a precise control situation, we should have defined, or if we want just grow the bacteria, we can use LB or undefined. After that, if we want to understand that how many bacteria, how many cells we have, we can use a spectrophotometer. we can use a spectrophotometer in the wavelength of 600 nanometers. We can use the spectrophotometer instrument. It will give us OD. 1 unit of OD presents this amount of cells per milliliter. If it's enough for us, we can go to the next step. Okay? Thank you. Fatima, I have so many emails and I have so many students. Sometimes I can't answer all of my emails. Okay, please, if you have any questions according our lessons, ask me in the class. I will answer you. Okay? If I have time, I will answer your email, but maybe sometimes because of so many emails, maybe it skips. Okay? So, what is the next step? What is the next step? The next step is to centrifuge. Look, this is the bacteria. Okay. This is the bacterial culture. Then we centrifuge it at 8,000 RPM at 8,000 RPM for 10 minutes. Okay, after that we will centrifuge the bacteria. I told you the bacteria are heavy and they will deposit in the bottom of the microtube. Yes, we have plenty of bacteria. What should we do with the supernatant? What should we do with the medium here? What should we do with the supernatant here? Tell me. I told you. No, I mean, what should we do with the supernatant? Here. We want what? We want bacteria, yes? We want here. We want the bacteria. Do we want the supernatant? The liquid here? No. What should we do? Remove them, dispose them. Yes, Nata. Thank you. We should dispose the supernatant. Thank you. Okay. The next step is to break the cell wall. Is to break the cell wall. You can see, break the cell walls. Disrupt cell membrane. So we have cell wall and cell membrane, and we should what? We should break both cell membrane and cell wall. How can we break the cell membrane? Do you know? How can we break the cell membrane? Do you know? Please tell me. How can we break the cell membrane of a bacteria? by centrifuge, not only centrifuge, Mr. Haidar. No, not antibiotic, kill the bacteria. We just want to break the cell walls. What else? We have two methods, physical methods and chemical methods. Okay, we can break them by physical methods and chemical methods. Okay, for example, in physical methods, the cells can disrupt by mechanical forces. For example, by some devices. Okay, no problem for them. Please give them the record class. Okay, the record class, and please tell all of the students. Okay, tell all of the students to be active in the class and study the book. Okay, for the next session, all of you, please study the book up to now. Okay. Please remind me. We have the lesson up to now in the next session. Okay. Please remind me. We are going through physical and chemical methods. We stop here. And who is the representative? Please, the representative